In this video, we're going to examine the effect of adding a catalyst on an equilibrium system. Let's consider a sample reaction here, the catalyzed decomposition of hydrogen peroxide into water and oxygen gas right here. Now again, normally a reaction like this will happen spontaneously, but the process happens very slowly unless we add in an iodide catalyst, which actually speeds up this reaction like this. Now remember that catalysts don't actually change the concentration of any of the reactants, and we're not changing the kinetic energy of any of the reactants the way we do if we would change temperature. Instead, catalysts such as iodide in this reaction here work by decreasing the activation energy of the reaction, which makes it easier to form the activated complex. Now, if we redraw the delta H of this reaction here with a catalyzed reaction, we can see that the EA has been lowered. Now, remember that you can measure both the forward EA and the reverse EA, but because of the difference that we see here, we see that we have a new EA of the forward reaction, and we also have a new activation energy of the reverse reaction, and we see that the magnitude of that decrease is the same in both cases. Now, if the magnitude of decrease for both the activation energy of the forward and reverse reactions is equal, that must mean that the effect on the rates of both the forward and the reverse reaction is also going to be equal. Now, what this means is that in an inorganic reaction like this with an inorganic catalyst, which is just a chemical we can add in order to make this reaction occur more easily, what these catalysts do is that because the magnitude of decrease in EA of both the forward and reverse reactions is the same, they speed up forward and reverse rates equally. Now, if this reaction is already at equilibrium, that means that there is going to be no change in the equilibrium because you're not changing the reaction in any way that would make one rate greater than the other one, thereby disrupting the equilibrium. So what this means is that adding an inorganic catalyst like iodide would cause a system at equilibrium to stay at equilibrium, but just that it would reach the equilibrium faster because, again, the catalyst accelerates the rate of a chemical reaction. Now, normally this would be a pretty boring video if I were to just say a catalyst doesn't do anything to change an equilibrium, because there are always exceptions to the rule, and those exceptions are what we call enzymes, which are your body's biological catalysts, usually made of protein molecules. Now, biological catalysts function in a fundamentally different way than inorganic catalysts do, in that they have a special structure called an active site, which is actually the location where the reaction's catalysis takes place. Now, the way that an active site works is that it has a specific shape and usually specific charges that allow it to attract only specific reactants in order to enter the enzyme. Now, what this means is that if a molecule doesn't match the shape and the charge of the active site, that means that a compound that doesn't match these two things would not even be able to enter the active site, and therefore no catalysis would take place at all. Now, if we consider two reactions that are opposite of one another, biology students will recognize this as the first step in cellular respiration, in which we want to take a phosphate from this large molecule here and put it onto glucose. In the reverse reaction, we want to break a phosphate off of glucose and put it back on this large molecule here. Now, obviously, you can see that the structure of the reactants and the products are very different from each other. Not just the structure, but also the charges as well. Remember that phosphate has negative charges. So, in the reverse reaction, glucose, the most common sugar in the body, would actually have a negative charge, but in the forward reaction, it would not. So because the reactants and products in biological reactions have radically different structures, this means that the forward and reverse reactions of biological reactions would actually require separate and opposite enzymes to do. And again, we can see this in the forward and reverse reaction of adding a phosphate ion to glucose. So the enzyme that is used to add phosphate to glucose is an enzyme that we call kinase, and again, its job is to break a phosphate off of this large molecule here and put it onto glucose, which means that the enzyme's active site is probably going to have some sort of positive charge that matches the shape of this large molecule here. But in the reverse reaction, the negative charge there is a negative charge rather on glucose, meaning that the active site that holds glucose in it is now going to have to be different to account for this negative charge, which is why the enzyme that does the reverse reaction is an enzyme called phosphatase. The names of these enzymes are not so much important. What is important is understanding that because enzymes rely on a specific active site with a specific shape and charge, they will only allow certain reactants to fit inside, and if any other reactants that don't match those enter, the enzyme doesn't work the way that it's supposed to, and therefore biological catalysts do actually have the ability to alter equilibria, because adding an enzyme can speed up the rate of catalysis in one direction while having zero effect on the other side, because the other enzyme requires a different active site. In the next video, we're going to look at how to represent changes to an equilibrium graphically and look at the process on how an equilibrium is achieved after it is disrupted by a stimulus.